Hello and welcome. This is the second part of this lecture about the membranes. This is a diagrammatic representation of the membrane structure. The membrane lipids are represented in blue color here. There are different types of lipids which are present in the membrane, as we have talked about that earlier. There are phospholipids, sphingolipids, glycolipids. In addition to the lipids, there are different types of proteins which are associated with the membranes. The membranes are shown in orange color here. There is a water both on the outside of the cell and on the inside of the cell. So the water molecules, they can interact with the polar head groups of lipids but are unable to solvate or interact with the nonpolar fatty acyl chains of the phospholipids. So therefore the membrane lipids are going to pack into this bilayer structure so that the polar head groups are going to face outside towards water, and the nonpolar fatty acyl chains are projected towards the interior of the membrane where there is no water molecule. So water is excluded from the interior of the membrane. Different types of carbohydrates are present. They are conjugated to different molecules on the exterior of the proteins. We talked about glycolipids. So these are the lipids which are conjugated to different types of carbohydrates. For example, the blood group antigens present on the surface of the red blood cells. Also, we talked about glycoproteins. So, for example, this is a protein to which the carbohydrates are attached on the exterior of the cell. So, this is a glycoprotein. So, different types of proteins are present in the membrane. Certain proteins, they are embedded in the membrane in such a manner that they pass through the membrane. Such proteins which pass through the membrane are called as the integral membrane proteins. This is another integral membrane protein. Another one is shown here and here. Many of the transport proteins, they form channels in the membrane. They have hollow interior through which different substances can move across the membrane. Such proteins are channel forming proteins. These proteins which form passages or channels within the membrane, they all are integral membrane proteins. While some other proteins are associated with the membrane in an indirect fashion. They are not directly embedded in the membrane, rather they're linked to some integral proteins as represented here. For example, this protein shown in yellow color is interacting with this integral membrane protein. And through this interaction, this protein is associated to the membrane indirectly. Such proteins which have an indirect association with a membrane, they're called as peripheral membrane proteins. They localize with the membrane, they're associated with the membrane, but they themselves are not embedded in the membrane. Other types of proteins, they are called as lipid-linked proteins. These proteins, they have a lipid molecule which is hydrophobic and it is covalently attached to this protein. These proteins are inserted into the membrane by means of this lipid which is linked, which is associated to this protein. Such proteins are called as lipid-linked proteins. These proteins are also associated with the membrane. So the integral proteins, they pass through the membrane. Since they pass through the membrane, so the region of this protein which is embedded within the membrane is hydrophobic. We will study about the nature of amino acids later on. Proteins are made up of 20 different types of amino acids. Some of these amino acids, they are non-polar. They are hydrophobic. So typically, those hydrophobic or non-polar amino acids are present in this part of the protein which is embedded inside the membrane. Because the interior of the membrane is hydrophobic because of the presence of the fatty acyl chains which are non-polar. Also, cholesterol molecules are present in the membrane. So the region of the protein which is passing through the membrane is rich in nonpolar hydrophobic amino acids. Biological membranes are asymmetrical. You would have studied about the cell membrane in cell biology, that the membrane is fluid as well as it is mosaic. The mosaicism in the membrane arises because of this non-symmetrical nature of the membrane. If you were to quickly look at the structure of the membrane on this outer surface, you can see that the proteins and the lipids are heavily glycosylated. They contain many different types of carbohydrates which are attached to them, both to the proteins as well as to the lipids. But you do not find any glycosylation of the membrane on the inside of the cell. Also, the proteins which are associated with the membrane, their distribution is also different on both of these regions of the membrane. This monolayer of the lipids is called as the outer leaflet of the membrane. This is the part of the membrane bilayer which is facing the exterior. This portion is called as the inner leaflet of the membrane because it is facing the inside of the cell. So the distribution of proteins in these various leaflets of the membrane is also different. Not only the localization of proteins is different, but also the lipids which are present in the membrane, they're different. The lipids which are present on the outer leaflet of the membrane and the inner leaflet of the membrane, they differ in terms of their composition and distribution. So therefore, biological membranes are asymmetrical because the distribution of proteins, lipids, and their conjugates are not the same in both of the leaflets of the membrane. This is a chart which is representing the percentage of different types of lipids which are present in the membrane. So the blue part here shows the inner leaflet of the membrane and the pink part represents the outer leaflet of the membrane. So this is the total amount of lipids present in the membrane. So you can see that sphingomyelin and phosphatidylcholine, they are more prevalent in the outer leaflet of the membrane as compared to their abundance in the inner leaflet. And here you can see that phosphatidylethylamine and phosphatidylserine, these are the more prevalent phospholipids which are present in the inner leaflet of the membrane. So the type of the lipids present in these leaflets of the membrane, their distribution, the type of the proteins which are present on the outer and the inner face of the membrane, they all are different. As a result of which, the membrane is going to be asymmetric in nature. This is a diagrammatic representation of integral membrane protein. So you can see here that this protein is passing through the membrane. It is embedded in the membrane. So this is an integral membrane protein. Some of the integral membrane proteins, they have one region which is embedded in the membrane. Some proteins can have more than one portion which is embedded in the membrane. The lipids of the membrane are shown in blue color, while the protein is represented in purple color here. So this is the outside of the membrane and this is the inside. So you can see that for this protein, some portion of the protein is embedded in the membrane. This part of the protein which is embedded in the membrane is rich in hydrophobic amino acids. The other protein represented here has 4 different regions which are passing through the membrane. So this protein passes through the membrane 4 times. All these regions which are passing through the membrane, or in other words, are embedded in the membrane, they are rich in hydrophobic amino acids. However, those portions which are on the exterior or the ones which are on the interior of the cell, since they're present in an aqueous environment, and water being a polar solvent is solvating these portions on the outside as well as on the inside. So the structure of the proteins is such that polar amino acids or hydrophilic amino acids are present on these regions on the interior and the exterior of the cell. However, the distribution of amino acids is such that the membrane spanning parts of the protein, they are rich in nonpolar hydrophobic amino acids. So some proteins can have a single portion of hydrophobic amino acid which is embedded in the membrane, while other proteins can have several patches of hydrophobic amino acids which are embedded in the membrane. Many of the membrane proteins, they contain the membrane spanning region as alpha helix. You would have briefly studied about alpha helix in some of the introductory courses. The other secondary structure element present in proteins is a beta sheet structure. Many of the proteins which are embedded in the membrane, they contain alpha helical regions which are found in the membrane. However, there are certain other types of proteins which contain beta sheet structures in them in the region which is passing through the membrane. So we've just talked about this, that there are hydrophilic as well as hydrophobic amino acids present in the membrane. The part of the protein which is on the exterior and on the interior of the membrane, it is rich in hydrophilic amino acids because these regions of the protein are present in an aqueous environment, and water is a polar solvent. So hydrophilic amino acids are abundantly present in these regions of the protein. However, the membrane-spanning part of the protein, since it is present in the membrane, it occupies a position in the hydrophobic part of the membrane. So this membrane-spanning part of the protein is rich in hydrophobic amino acids. There is a very important computational technique which could be used to infer or detect that whether a given protein is a membrane protein or not. And if it is a membrane protein, which part of the protein is expected to be embedded in the membrane. This computational mechanism is based upon the free energy of transfer of amino acids from the membrane to water. If there are polar amino acids, the stability of polar amino acids within the membrane will be quite low. So the free energy of polar amino acids in the membrane will be very high. But since they're polar, so when they will be transferred to water, the free energy will decrease and the system will become more stable. On the other hand, the non-polar amino acids, they will be more stable within the membrane because the environment around them is also non-polar. And if these non-polar amino acids are transferred to water, then they will become unstable and the free energy will be more. So this computational method is based upon the free energy of transfer of an individual amino acid from the membrane to water. Hydrophilic amino acids, when they will be transferred from membrane to water, they will now become stable, and hence the free energy change will be negative. On the other hand, if there are hydrophobic amino acids, then the transfer from the membrane to water will be energetically less stable. So therefore, based upon this mechanism, we can determine which part of the membrane is embedded, which part of the protein is embedded in the membrane. The thickness of the membrane is such that 20 amino acids forming a alpha helix are going to span through the membrane. So in order to determine the portion of the protein which is expected to be embedded in the membrane, what we do is that we look at a series of 20 amino acids together and we look at their potential of transfer from membrane to water. This is a diagrammatic representation showing a membrane embedded protein. This portion is the lipid bilayer. This part of the protein is embedded in the membrane. This is the region of the protein which is on the outside of the cell membrane, and this is the part of the protein which is present on the cytoplasmic phase of the membrane. And these are the amino acids which are embedded in the membrane. Also keep in mind that the thickness of the membrane is such that it roughly accommodates about 20 amino acids. So what we do is that we look at the 20 amino acids, a window of 20 amino acids, say for example amino acid number 1 till amino acid number 20 here, and we add the free energy of transfer from the membrane to water for all of these individual amino acids, from amino acid number 1 to amino acid number 20. That is represented as the first point on this graph. Now what we do, we look at the next window of 20 amino acids. The next window will start from the second amino acid to amino acid number 21. So from here till here, we add up the free energy of transfer from membrane to water for all of these 20 amino acids, that is amino acid number 2 to amino acid number 21. We add those numbers and we plot that on this graph. Then the third window will be amino acid number 3 till amino acid number 22. So we add the free energy of transfer for all these 20 amino acids, and that is shown as point number 3 on this graph. In this way, we keep on looking at the free energy of transfer for a window of 20 amino acids. The next window will be from amino acid number 4 to amino acid number 23. So we add the free energy of transfer of these 20 amino acids, and that will be the fourth point on the graph. The next one would be amino acid number 5 till amino acid number 24. So we add the free energy of transfer of these 20 amino acids, and that will be the point number 5 on this graph. So in this way, we are going to look at the cumulative effect of 20 amino acids together. Since the thickness of the membrane is such that it can accommodate 20 amino acids in form of an alpha helix, so we look at the region of the protein which is going to be most hydrophobic. If a segment of any given segment of 20 amino acids contain polar amino acids in it, then the free energy of transfer from membrane to water will be quite low, because these are mostly polar amino acids and hence they're going to be more stable in water. But if we have non-polar amino acids, consecutive 20 non-polar amino acids, or most of the 20 amino acids being non-polar, so that means that their free energy of transfer to water is going to be extremely high, because there are going to be very little stability in water. So therefore, by looking at this combined effect of 20 amino acids, we can find a window, we can find a region of the protein which is going to be most hydrophobic. So the part of the protein which shows the maximum free energy of transfer from membrane to water, that is going to be regarded as the most hydrophobic part of the protein. In this graph represented on the right, a window of 20 amino acids is plotted on the x-axis, and the free energy of transfer from the membrane to water is shown on the y-axis. So we plot this change of free energy in terms of a window of 20 amino acids. So on the x-axis, the first amino acid of the window is represented. So we see that this entire portion of the protein up to around 70 amino acids is present on the outside of the membrane, and hence contains more hydrophilic amino acids in it. And this is represented in the graph here as well, that you do not see a high amount of free energy when this portion is going to be transferred into water. That means this part of the protein is more stable in water. Hence, it is expected to have more hydrophilic amino acids in it. But as we approach close to amino acid number 73, which is shown here, so as we are going to move towards this part of the protein, you can see that there is a significant increase in the free energy of transfer from the membrane to the water. This means this transfer is energetically unfavorable. So this is energetically unfavorable because this part of the membrane close to amino acid number 73 and is hydrophobic or non-polar. That is why when it is being transferred to water, it is showing a very high value of free energy. That means it has a very little stability in the aqueous environment. And this little stability in the aqueous environment is because of the presence of many non-polar amino acids within this portion. So based upon the computational programs, we define a cutoff value. Those regions of the protein which have a free energy or higher free energy transfer value than this threshold are attributed to be the membrane embedded regions of the protein. So that means amino acid number 73 is going to be the first amino acid of this window which is going to be embedded in the membrane. And as we move forward, we move on to the region of the protein which is present inside the cell. So again, this part of the protein is rich in hydrophilic amino acids. So you see that the free energy of transfer is quite low. That means, again, this portion, when being transferred to water, has a high stability in water. Therefore, the free energy change is quite low here. So in this way, by computationally looking at the free energy of transfer from a nonpolar to a polar environment, which is water, we can find that which regions of the protein are going to be most unstable. And the regions which are most unstable in water are the parts which are most hydrophobic. And since they have a certain degree of hydrophobicity which is more than the cutoff value, so therefore they are expected to be embedded in the membrane. By using this mechanism, we can computationally predict about the structure of the proteins, that which parts of the protein are expected to be embedded in the membrane. In this representation of the graph, you can see that there is only one portion which has sufficiently high hydrophobicity to be embedded in the membrane. So this protein is expected to have only one transmembrane region. The part of the protein which is embedded in the membrane, that is known as the transmembrane region. Since this is a helix, so this is called as transmembrane helix. Some proteins, on the other hand, they can have several transmembrane regions. For example, the one shown here contains 4 transmembrane helices. So when you're going to look at such a plot for this protein, you will find 4 regions of this protein which are going to be above this threshold value, above this cutoff value. So if a protein contains only one transmembrane region, it will show one peak above the threshold. But if a protein contains many hydrophobic regions which are embedded in the membrane, then that protein will show several peaks above this cutoff. So by counting the number of the peaks which are above this threshold, we can find the number of transmembrane helices present in a given protein. This next figure represents the structure of cytochrome B5. Cytochrome B5 is a protein which is embedded in the membrane. Here you can see that this part of the protein is hydrophobic. It is associated with the membrane, and this portion of the protein is rich in hydrophilic amino acids. Some proteins are attached to the membrane by means of a covalently bound lipid on them. These are called as lipid-linked proteins because they're attached to the membrane by means of a covalently bound lipid to them. One of the type of these proteins are called as prenylated proteins as they contain prenyl units in them. The prenyl units are made up of 5 carbon units which are called as isoprene. Different types of isoprene units assemble with each other to form other types of prenylated units. For example, 3 isoprenes attached to each other, they form farnesyl residue. This farnesyl portion is then attached to the protein. This is a 20 carbon residue which contains 4 isoprene units in it. This is called as granyl-granyl residue, and this carbon is in turn linked to the protein. So these proteins are inserted into the membrane by means of these nonpolar portion of the isoprene units. Sometimes this part is also called as the lipid anchor because this part is going to anchor the protein into the membrane. The other type of lipid anchors, they're called as glycosyl phosphatidylinositol. So this portion shown in green color is phosphatidylinositol. So this is a glycerol, carbon number 1, carbon 2, carbon 3, and these are two fatty acids attached to glycerol. This is a phosphate linked to the third carbon of glycerol to which an inositol is attached. So this is a phospholipid. This is phosphatidylinositol. With this phosphatidylinositol, there is a tetrasaccharide, a carbohydrate moiety which is attached, and then through this extension, it is going to be linked to the protein. So these proteins are interacting, they are covalently attached to this phosphatidylinositol molecule, and these fatty acids of the phosphatidylinositol are going to be embedded in the membrane. So the integral proteins, they themselves are inserted into the membrane. The peripheral protein, they interact with the surface of the membrane. They're not embedded in the membrane, rather they interact with the surface of the membrane through other proteins. And the third type are the lipid-linked proteins, which contain a lipid covalently attached to them, and by means of that nonpolar lipid, they are embedded into the membrane. So next we're going to look at the membrane of a red blood cell, the erythrocyte membrane, as a model. This is a diagrammatic representation showing the structure of a red blood cell. So the membrane has been sliced here, and the membrane is now opened from this part of the cell. So this portion, which is facing towards you, is representing the inner leaflet of the membrane. So this is the cytoplasmic phase of the membrane. And the one on the other side, which is away from us, that is the outer leaflet of the membrane, or the part of the membrane which faces the outside. So you can see many different types of proteins which are associated with the lipids bilayer. The lipid bilayer is represented in blue color. One of the important proteins which is represented here is a cytoskeletal protein called spectrin. Spectrin forms filaments which are associated with the lipids, and therefore they are important for regulating the structure of the cell. The next one represented here are actin proteins. Actin are also cytoskeletal proteins, and they're important for regulating the shape of the cell. That's another important protein called tropomyosin, which is important for regulating the structure of actin and in turn governs the shape of the cell. This protein, which is the so-called band 4.1 protein, that is a protein which mediates interaction between spectrin and actin. Both actin and spectrin, they're cytoskeletal proteins, and they're important for regulating the shape of the cell. They're also important for the attachment of other types of proteins to the membrane by means of the cytoskeleton. This protein is anchorin. The protein anchorin is important for interaction between other proteins and the membrane. This is a protein which is so-called band 4.2. This protein is also important for regulating the shape of the cell. This is the protein glycophorin A. The protein glycophorin A has several roles to carry out. One of the functions is that this is an antigenic protein, and it contains the antigens for MN blood groups. ABO blood group system is the most prevalent type of blood groups, but in addition, based upon several other different types of antigens, there are other blood grouping systems as well. One such blood grouping system is called as the MN blood groups based upon the MN antigens. So it is the type of the glycophorin which govern this type of blood groups, M or N. This is an anion transport channel protein. This protein is important for the transport of anions. Anions are negatively charged ions. This protein is typically very important. Well, there are several different types of anion channel proteins, and these proteins they are typically very important for the function of the red blood cell. You would have studied a little about this anion transport channel in cell biology as well. Here we are going to have a quick look at the function of this transport protein. In this diagrammatic representation, you can see that this is a red blood cell. This is the interior of the red blood cell. The blood cells are present in blood, so outside is the blood plasma. And this is the internal region of the red blood cell. Red blood cells, they carry oxygen from the lungs to other parts of the body. Red blood cells not only transport oxygen, but they're also very important for the transport of carbon dioxide. There are several ways for the transport of carbon dioxide. Some carbon dioxide is transported as a gas, as carbon dioxide gas. But this is a very small proportion of carbon dioxide which is transported as gas through blood. Much of the carbon dioxide is transported in form of bicarbonate ions. Carbon dioxide can react with water and therefore it produces carbonic acid. Carbonic acid decomposes into bicarbonate ions and hydrogen ions. Much of the transport of carbon dioxide is done by means of converting that carbon dioxide into bicarbonate ions. Some of the carbon dioxide is also transported through hemoglobin. There is an iron atom in hemoglobin which can bind to either oxygen or carbon dioxide. So some carbon dioxide is also transported by hemoglobin as well. When the red blood cells they take up oxygen from the lungs, they move to the other parts of the body. So when the red blood cells they reach the body cells, they reach the body tissues, they give away their loaded oxygen and take up carbon dioxide from the surrounding tissue. When this carbon dioxide diffuses into the red blood cell, it is converted into carbonic acid by this enzyme called as carbonic anhydrase. The enzyme carbonic anhydrase carries out a reversible reaction. It depends upon the relative concentration of carbon dioxide and bicarbonate ions. In the tissues, oxygen is given away. The tissues are carrying out respiration, so they produce carbon dioxide. This excess carbon dioxide diffuses into the red blood cell where the enzyme carbonic anhydrase, since the concentration of carbon dioxide is greater, so the forward reaction is going to be favored. Hence, this carbon dioxide is going to be converted in form of bicarbonate ions. These bicarbonate ions are then given out into the blood plasma. So the net result is that carbon dioxide, which was a gas produced by the cells through respiration, is eventually going to be converted in form of bicarbonate ions. Then these bicarbonate ions are present in the blood plasma, and through blood circulation, they are taken to the other parts of the body, to the lungs. Now, when the bicarbonate ions they go out of the red blood cell, a chloride ion is going to be pushed inside. This type of transport you would remember from cell biology is called as antipode, in which substances are going to be transported in the opposite direction. So this movement of ions is important to maintain the electrical neutrality inside the red blood cells. Because if bicarbonate ions, which are negatively charged, are being constantly pushed out of the cell, then the protons are going to be accumulated in this red blood cell. Hence, the red blood cell will have a large excess of hydrogen ions. And if the hydrogen ions concentration increase, that results in producing an acidic environment. So the pH will be decreasing. In order to counter this excess of protons, or in other words, the hydrogen ions, the chloride ions are going to be pushed into the red blood cell. So therefore, these chloride ions are going to maintain electrical neutrality with these hydrogen ions here. Now, when the red blood cells are present in the lungs, the concentration of oxygen is more here. So oxygen is going to diffuse into these red blood cells. The red blood cells contain carbonic anhydrase. Now, since the concentration of carbon dioxide is less here, the partial pressure is less, so therefore carbon dioxide is going to diffuse out of the cell. When carbon dioxide is going to diffuse out of the cell into the alveoli, the reaction is going to take place in the reverse direction because one of the reactants is going to deplete here. So enzyme carbonic anhydrase now carries out the reaction in the opposite direction, and thereby it is going to convert bicarbonate ions into carbon dioxide, and this carbon dioxide is going to diffuse out into the alveoli. So therefore, the concentration of bicarbonate will decrease, so more bicarbonate is going to be pumped into the cell. So therefore, in order to accommodate for that incoming bicarbonate, chloride ions are going to be pushed out of the cell. So the anion channel is very important in regulating the movement of the anions across the red blood cell membrane, and therefore is very important to regulate the pH and the electrical neutrality of the red blood cell. Again, coming to this figure, so the anion channel is represented here. This is important for the transport of anions, and this transport of anions is very important with respect to the function of the red blood cells to transport carbon dioxide. So anion channel is important for regulating the pH and the electrical neutrality of the erythrocytes. So this is again something we have commented about earlier as well while talking about carbohydrates. This is about the ABO blood groups. The ABO blood groups are the antigens which are present on the surface of the red blood cells. So these are specific type of glycolipids which are present on the red blood cells, and the carbohydrate part, so this is the base oligosaccharide. If N-acetyl galactosamine is attached to it, that is A type blood group. And if to this core oligosaccharide galactose is added, that is the B type. So those people who have AB blood group, they have got both of these glycolipid antigens on the surface of their red blood cells. The blood group antigens are not the only type of carbohydrate present on the surface of the red blood cell. Rather, there are many other different types of proteins which contain carbohydrates attached to them. This is an electron micrograph of a red blood cell, and this is the cell membrane shown here by this thick boundary. So this is the outside of the cell, and this is the interior. On the outside of the cell, you can see a lot of structures projecting outward on the exterior. These are all different types of carbohydrate moieties which are associated, which are attached to different lipids and proteins on the surface of the red blood cell. Collectively, this entire structure of carbohydrates surrounding a given cell is called as the glycocalyx of that cell. These carbohydrates which are present in the glycocalyx are important for many different functions. One such function is, for example, in case of the red blood cell, is the blood groups. The blood group antigens are governed by the type of the oligosaccharide. The attachment of the cells is also mediated by different types of carbohydrates and their respective binding proteins, and those carbohydrates are also a part of the glycocalyx of that cell. So in terms of recognition, cell signaling, and communication, and determining and controlling several other functions of the cell, these carbohydrates present on the surface of the cell are quite important. Although the cells are isolated, but some cells, they have a direct route to transport substances through them. Such routes are called as the gap junctions. This is a diagrammatic representation showing the cell membrane shown in blue color of one cell, and this is the cell membrane of another cell. So these are two different cells. This is the cytoplasm of the second cell. This is the cytoplasm of the first cell. If substances are to be transported, they should pass through the cell membrane of the first cell, go outside, and then should be transported through the cell membrane of the second cell. Now, this is going to be energetically costly. So, what some cells have a mechanism that they have special sets of protein. These proteins are channel forming proteins, and they are going to connect the membrane of these two adjacent cells. And they form a junction. This junction is called as the gap junction. The gap junction is a continuous pore extending from one cell to the other cell. Certain selective type of substances, mostly small substances like ions, amino acids, sugars, etc., or some other small molecules, they can be directly transported from the cytoplasm of a cell to the cytoplasm of another neighboring cell. Large molecules like proteins and nucleic acids, they cannot move through this channel. So a gap junction is a continuous channel formed by proteins present on the surface of two adjacent cells.